Video we are going to discuss about the lock and key model of enzyme mechanism. This model was proposed by Emil Fischer in 1898, and this model is also known as template model. Now let us discuss this mechanism in detail. This is the lock and key model. Here you can see an enzyme. This is the enzyme. Here, this is the active site and these are the substrate. So, the active site means the site at which the substrate binds on an enzyme. That is, the site on an enzyme at which the substrate binds is known as active site. And this active site is specific. It is specific. Specific means it accepts only the specific substrate, not all the substrates. Okay, it binds only on specific substrate. That is, it is known as the active site is specific. So the enzyme binds to the specific substrate. Okay, that is the substrate binds at the active site of the enzyme and forming enzyme substrate complex. And this substrates then converted to product by the enzyme. So we get enzyme product complex. And at last, the products are released from the enzyme and the enzyme is reused for another round of reaction. This is the lock and key model. As the name is lock and key because this enzyme and substrate binds like a lock and key. That is, the enzyme acts like a lock and the substrate like a key. So the enzyme and substrate bind and forming enzyme substrate complex like a lock and key. So the name is lock and key model. Now let us discuss in detail. Binding of the substrate and the enzyme takes place at the active site in a manner similar to the one where a key fits a lock and results in the formation of an enzyme substrate complex. Then that is substrate and enzyme binds forming enzyme substrate complex, and this binding is like a lock and key. Then the next point is the enzyme substrate complex formed is highly unstable. So they immediately decomposes to produce the end product of the reaction and to regenerate the free enzyme. That is, when the enzyme substrate complex is formed, it is highly unstable and it has the tendency to decompose immediately and to give products and then regenerate the free enzyme. However, this model has some drawbacks. That is, it cannot explain the stability of the transitional state of the enzyme and also the concept of the rigidity of the active site. This model could not explain the concept of the rigidity of the active site and about the stability of the transitional state. So this model cannot be accepted. So the lock and key model means the enzyme and substrate binds like a lock and key and forming the enzyme substrate complex, and then it decomposes immediately and forming product and the enzyme is released. This is all about lock and key model. Thank you.